Good morning, everybody. We're really excited to welcome you to our West Hour webinars, Empowering Science and Technology. This morning, we have Dr. Ingram Vargas, who's one of our local field application scientists. He's going to present to you developing scalable RNA-LNP drug products for clinical readiness. He's based in San Diego, California, and covers the Southwest Territory. Ingram received his PhD in material science and engineering from the University of California, San Diego. So we're real excited for you to get to know Ingram better, and a great way to do that is to hear his presentation. So before we begin, we do just have some housekeeping. So for those that don't know, the West Territory is divided into two territories, the Southwest and the Northwest. So let's start with the Northwest. We have Angel Galarza, who's the Regional Sales Director for the Northwest Territory located in San Francisco. And then we also have Ian Biamania, who is a Senior Field Application Scientist located in Seattle. Ian isn't here today, he's on paternity leave. For the Southwest team, we also have Viet Nguyen, who's a field application scientist who also happens to be on holiday. And then obviously we have Ingram Vargas, who covers the Southern California territory all the way out to Texas, so west. And then I'm Kim Killian, I'm the Regional Sales Manager for the Southwest Territory located in San Diego. Jason Coleman has been so kind to show up for us today. He represents the East Coast and he manages all of the field application scientists. So we're really excited and appreciate him showing up for us today. You know, just a reminder, this is, I think, our second year of doing webinars. If you're new to our webinars and you're curious about past webinars, you can go on to YouTube and find our playlist, and you can locate them under West Hour. And then finally, you know, just a reminder, we do have journal clubs, usually the second Friday of every month. So stay tuned and we'll be sending you out information regarding those journal clubs. So I think that's everything. Thanks again for your participation. And Ingram, I will hand it off to you.
Okay, thanks, Kim, for the introductions. And hello, everyone. As Kim said, my name is Ingram Vargas. And today we were going to talk about developing scalable RNA-LNP drug products for clinical readiness. So we always like to start with our vision, which is to accelerate the creation of transformative medicine that significantly impacts human well-being. And with this, we work with world-leading drug developers to understand different diseases and create therapeutics and vaccines that define the future of medicine. We are a global leader in nanoparticle solutions with headquarters in Vancouver, Canada. And currently, we are building a new facility that will include multiple GMP suites for manufacturing RNA therapeutics and vaccines. And what we offer at Precision NanoSystems is these three main pillars or platforms. The first one is the nanoassembler manufacturing platform, which are our instruments. We also offer the Genboy delivery platform, which are lipid mixes to produce the lipid nanoparticles. And we also offer the genetic payload platform, which currently we are developing proprietary self-amplifying RNA to express different proteins, including antigens using RNA vaccines against COVID-19. So we offer end-to-end manufacturing solutions for genomic medicines. And while at PNI, we focus on the self-assembly of the nanoparticles, the instrument, and the lipids. Our partners at Cytiva and Paul offers different technology regarding tangential flow filtration, sterile filtration, and fill and finish. this to end up with a drug product. So as we all know, genetic medicines are the future. They allow us to target any gene in any way, and their versatility allows us to silence, express, or edit genes. And which is more important is that these drugs are completely designed, which means that we have endless possibilities of the medicines that we can create. Some of the most advanced methods to deliver nucleic acids to the cells is with lipid nanoparticles. Lipid nanoparticles are complex structures that can protect the nucleic acid and are formed by several components, which are the lipids. And the way that we manufacture this is with microfluidic technology. So the way the microfluidic technology works is by injecting two different reagents at the beginning of a microfluidic path. One containing the payload that normally is in an aqueous solution, and then the other containing a lipid mix that normally is in a solvent solution, most regularly is an ethanolic solution. So the reagents mix through the microfluidic pad or the microfluidic channel that is especially designed to generate a controlled time invariant mixing, which result in the formation of optimal lipid nanoparticles. One of the main advantages of using this architecture is that we use the same architecture for all our instruments, and this allows that the critical parameters of particle formation can be maintained across different scales. So at Precision NanoSystems, we offer solutions from research through commercial, and we come with several instruments for that. For example, when you're starting your genomic medicine program, we have the Spark, and this is going to allow you to screen through different formulations and choose your leads. And then from there, we can go to the Ignite and optimize the formulations. And most recently, we launched the Ignite Plus. That the advantages of the Ignite Plus over the Ignite is that we are going to be able to generate larger volumes of formulation and also higher flow rates, which will allow us to de-risk and extend the capabilities of our formulations by simulating a GMP system. And then after that, we have the Blaze. The Blaze will allow us to create or to formulate larger volumes that will allow us to explore process development, the process development aspects of the drugs. That could be, for example, tangential flow filtration. And then from there, we can go to the GMP system. And then one of the advantages is that all, as I mentioned before, all the instruments uses the same microfluidic technology. So since all the instruments use the same microfluidic technology, the scale-up and nanoparticle manufacturing becomes easier compared with other formation methods that use on steady mixing conditions. For example, in our microfluidic architecture, we use time invariant mixing, which means that every time that you perform a formulation, if it's with the same parameters, you will have a consistency in the results. And that at the same time allows you to scale up in an easier way. So for example, here, we have the Ignite, which will allow us to do formulation development and optimization of those formulations. And then we could go directly to the Blaze system, which will allow us to perform formulations with higher rates and higher volumes. And then from there, we can jump to the GMP system. But what we are offering right now is the Ignite Plus, that is going to fill those spaces between the Ignite, the Blaze, and the GMP systems. And with this, I mean that, for example, the Ignite Plus is going to help us have larger volumes at the Ignite. For example, the Ignite can go up to 20 milliliters, and the Ignite Plus can formulate up to 60 milliliters. But on top of that, one of the advantages of the Ignite Plus is that we can start having flow rates comparable to those of a GMP system. So that is going to allow us to do the transition from discovery to commercial in a more smooth way. So the workflow of the Ignite Plus looks like something like this. First, we load the cartridge. One important thing here to mention is that all our technology uses single-use cartridges. The cartridge contains the microfluidic pad, and since it's single-use, you will account for no contamination between your samples, since you can do one formulation per cartridge. So the way that it works is that you will introduce the cartridge into the Ignite Plus, and then you will load the pre-filled syringes with your reagents, in this case, the aqueous and the solvent reagents. And one of the advantages of the Ignite Plus is that you can load 2 30 milliliter syringes into the system. And compared with the Ignite only allows you to load 2 10 milliliter syringes. So this is what allows you to get higher volumes. And then when you load the pre-filled syringes, you will either create a recipe in the system, or you can choose one of the recipes that you have created before. After that, you have to load collection tubes, and then you will just start the formulation, and then at the end of the formulation, you can collect the sample. So one important thing to mention here is that the Ignite Plus has 2 different cartridges depending on the flow rates that we want to achieve within our formulations. The first cartridge is the next generation cartridge, that is the same one that the Ignite uses. And if we want to increase the flow rate in our formulations, we have now the next gen 500 mixer that it works the same. It just allows you to achieve higher formulation rates. So yeah, so to showcase, so with this technology at Precision NanoSystems, we are currently developing a self-amplifying RNA COVID vaccine, where the saRNA is first synthesized from a DNA template and encapsulated into LNPs. And then when the saRNA is delivered to cells, it produces multiple copies of mRNA and expresses the vaccine antigen. Some of the advantages of using self-amplifying RNA to produce vaccines instead of just mRNA is that it can be 10 to 100 times more potent, and that's the reason we are exploring this. So to showcase the capabilities of the Ignite Plus, we choose the saRNA COVID lipid nanoparticles as a model drug product. And in this case, we're going to study, what we studied was the different parameters that we can have within the same system and the results of that. So this means that we perform across different volumes and different flow rates. So what we have here is we perform 4 different scenarios. The first one with low flow, low volume. The second one with low flow and high volume. The third one is going to be a high flow, low volume. And the fourth one is going to be a high flow, high volume scenario. So the results show that we are able to maintain critical quality attributes of the lipid nanoparticles, such as size, polydispersity index, and encapsulation efficiency. Moreover, we show that we are able to maintain the lipid composition of the final products as well as the saRNA integrity. The volumes, the great advantage is that to generate critical process parameters, the Ignite Plus requires smaller volumes and simpler workflows than any other systems, resulting in the reduction of time, cost, and risk. So when comparing the effect of the lipid nanoparticles produced across the different volumes and different flow rates, we can observe that in vitro, the protein expression is maintained in a dose-dependent manner. So in here, with those, the cells with different doses of the lipid nanoparticles that we produce within the different scenarios. And we observe that all those nanoparticles give us similar results in terms of protein expression. What is more interesting is that when we test the lipid nanoparticles in vivo, we observe similar immune response within the different scenarios that we selected. And in this case, the immunization studies were performed in mice, where bleeds were collected 2 days prior to immunization, and on day 27, the immunization was administered via intramuscular injection on day 0 and on day 28. And the results presented here in this plot are correspond to day 42 after the second intramuscular injection. So the second intramuscular injection was the booster from the first vaccine, and these are the results. As we can see here, the different parameters, or the results across the different parameters that we explore are really similar, which tells us that we are able to de-risk the process at very low scales, and that is maintained through really high ones, for example, the high flow and high volume. So talking about process development, inlay dilution is one of the most important processes to study. And in this case, we are studying inline dilution when we are producing liposomes. It is important to mention that this study was done in liposomes and not in lipid nanoparticles, just because the liposomes are more sensitive to inlay dilution or bulk dilution than lipid nanoparticles. And we wanted to showcase was the implication of study inlay dilutions since the very first beginning. So inlay dilution becomes more important at larger scales since the formulation time can increase, and that can bring some issues for the particles that may start to degrade over the time. But since we are studying this since the very first beginning of our research, we are able to de-risk these situations. So this becomes something essential to accelerate timelines, reducing risk, and minimizing costs. In terms of solvent removal, as we mentioned at the very first beginning, solvent removal is one of the things that we need to perform after particle formulation. And this is because right after our formulation, we have the lipid nanoparticles suspended in an ethanolic solution. So to perform in vivo studies or in vitro studies, we want to remove that ethanol from the nanoparticles and replace it for some other solvent. So in here, we are comparing the different scenarios and then post formulation, post dilution, and then post sterile filtration. And we can observe that we can do that with the Ignite Plus, because the Ignite Plus offers to formulate volumes around 40 milliliters, which could be used to start process development in terms of tangential flow filtration. So this means that with smaller volumes, we can start doing this, which is going to allow us again to de-risk further studies. For example, like with the Blaze, instead of going like directly from a formulation that is 20 milliliters to a formulation that is 1 liter, I can stop in between them now and say, okay, like I don't want to go to really high volumes, so I want to go to a volume that is going to allow me to study tangential flow filtration. But at the same time, I want a volume that is lower than, for example, 1 liter, and that can come in terms of costs, risks, and also time. So in summary, microfluidics such as the next generation mixers enable consistent and scalable production of RNA-based lipid nanoparticles. And the NanoAssembler Ignite Plus can prepare volumes that are clinically relevant, like in terms of, for example, flow rates. So we can start mimicking what would happen in a GMP system from really early stages on our research, which will minimizes the risks to produce a drug based on lipid nanoparticles. And that's pretty much it. We want to acknowledge our collaborators and the different departments at Precision NanoSystems. So thank you so much for listening. I don't know if there's any questions.
I'll turn over you to say your thank yous, but I'll say my thank yous to everyone here, and thank you, Ingram. This was very insightful.
Okay, well, thank you everyone for joining.
Okay, thank you. We appreciate your time and your questions. If you think of another question after the webinar, feel free to reach out to Ingram and he can follow up, Jason and Ingram, or feel free to reach out to Angel or myself. We will get your question answered. If anybody's interested in continuing the conversations about Ignite Plus, again, too, don't hesitate to reach out to us. And we did record this, so if you want to send it to a colleague or you want to go over any information, you can find it on our playlist. So again, we look forward to seeing you in August for the Journal Club, and we hope you're having a safe and enjoyable summer, and we look forward to the next time we see you. So thanks again. Have a great day. Thank you. Bye-bye.